First, I wanted to just respond to some discussion that came up in the first talk of the conference on how do we speak to politicians. And I just want to say, I've learned over the last 20 years, they don't really find our organisms that sexy, but what they do care about is what they do and their function. And so I feel like a strategy to try to share with the public is really try to share the really valuable critical function that these soil organisms are performing. So I wanted to also just mention my co-author, Hannah Steele. She and I just met 3 years ago. We independently were doing research that fit together very nicely. And we met in Belgium and discovered this symbiosis, or the complementarity. So she and I are really equals in this presentation. Some people ask if she was my student. No, she was not. We did that. She's an independent scientist in her own right and a postdoc at University of Ghent. I also want to say a special thanks to two people that have been pivotal to these findings. And for me, I wanted to thank Noah Führer for hosting me during my sabbatical a couple years ago when I did the sequencing work with the microbial community in this, and for Hannah, she wanted to acknowledge Wim Bert, who was her mentor at Ghent. So composting is a heat-generating and anaerobic process of the degradation of organic matter and the final product being humus. Compost has been touted for its use in fertility, a means for processing organic waste, weed management, disease suppression, as well as promoting plant growth. But not all composts are equal. And that's part of why we see a lot of inconsistent reports in the literature. The biological communities of compost are greatly affected by the source of carbon going into them, the method of the process of composting, and the duration of the curing process, or the maturity of that. Compost is a successional process. The stages that I'll be referring to are on the lower portion of the panel, highlighted there with the blue. And so we see a succession in temperature, which really drives this, the decomposition rate. But through these different phases, we see very distinct changes in the biological community as well as the soil chemistry. Now, for economic reasons, many states, countries have set regulations to monitor and provide guidelines for the thermophilic phase. This is a phase where it's defined as having reaching temperatures of about 55 to 77 degrees Celsius for a minimum period. And the idea here is more of a sanitation to kill pathogens and weeds. This is very important, but very little attention is paid to the stage that follows that, which is a curing phase when the temperatures are cooling. So after we have that sanitation phase, really a lot of the beneficial attributes of compost occur after that thermophilic phase during the curing. And that's going to be the component of the process that we're going to really focus on in our presentation, because this is where the biocontrol organisms colonize, and this is where the complexity of the food web builds that can play a lot of important ecological roles. So the objective is to really focus on thinking of indicators. The current commercial composters are very interested in how to define when a compost is ready to use, when it's finished or cured or mature, but a lot of the existing metrics are very vague. They're based on like respiration or carbon to nitrogen ratios, simple microbial carbon. They don't tell us a lot, and there's a lot of variation. So we want to complement those in our studies by looking at specific community composition, and particularly looking at the composition of microbial communities, fungal and bacterial, and nematodes as biological indicators. And then I will also show an example of their compost use application in the field, for disease suppression. Now, regulations define what the carbon to nitrogen ratio should be to make proper compost to reach those thermophilic thresholds. Usually they're aiming for about 25 to 40 parts of carbon per part of nitrogen. And there's certainly, you need a certain amount of moisture and turning process to achieve those temperatures. However, there's nothing said about the source or the quality of carbon. So illustrated on this slide are a few of the recipes that I have tested, and you'll later see the data in the field study where we had specific recipes designed that met the carbon to nitrogen ratio and moisture requirements. The control in this case is a control based on where I come from in Vermont, where dairy is common, and dairy manure is a major source of nitrogen and a concern for organic waste management. And so the control is really what the farmers do. They just are putting out their bedding mixed with manure out the end of the barn, and so I call it a manure silage control. It's extra high in nitrogen in this case. But then I took the 3 sources of carbon and I put the relative ratios here so you can see. And I won't show you the data, but these do yield very different, significantly different bacterial and microbial, bacterial and fungal communities. There are different methods for the curing process. The traditional is using windrow, which is long, rows that get turned by a tractor. Sometimes there's equipment that can just go over it and flip it over to add air and keep the temperature rolling. But there's also a process that can take less land space and be a little faster, and that's called aerated static pile, where you're not turning the pile, but you're actually forcing oxygen up through the pile. Another approach has been, and so those are both always known as thermophilic heat generating. The third is vermicompost, which utilizes earthworms in the curing phase. Now, I'll mention earthworms, if you reach 55 degrees Celsius, you'd kill them. So if you only use earthworms and that's your entire composting process, that's mesophilic. But if you want to meet the standards for the sanitation, the way that operators are choosing to do it is start with an aerated static pile, reach the sanitation phase, and then have a second phase where they go through the vermiculture. So I wanted to show some data on the curing phase where I took a consistent recipe, replicated piles, and then it all began as a treated through aerated static pile technique and then was cured by 3 different methods. By the windrow, the aerated static pile, and the vermicompost, followed by the aerated static pile. The yellow line or row there is days of composting. If it's white, the 22 and 56 degrees, that's still the thermophilic phase. If it's yellow, that means it's post-thermophilic and in the mid to late curing. This style of graph is called a heat map. So the red, it means it's highly abundant. White is intermediate, blue is low abundance. These are percentages of sequences representing these taxa. So at the end of the thermophilic phase, one of the things we see highlighted here, and it follows what we would predict, are bacteria that are thermophilic. They're known thermophiles. So we see a predominance of the alpha proteobacteria, especially in the aerated static pile and the vermicompost, and the firmicutes. And firmicutes are known, well-known thermophiles. If you go later in kind of the mid to late curing phase, we see a shift in that community composition, and now the bacteroidetes become quite dominant. These are, quite prevalent in manures, so it's not surprising. And the actinobacteria also become relatively abundant. So as Noah Fearer was talking about kind of the oligotrophs, I want to say we're seeing this kind of trajectory in compost succession. Early in the phase, it's more of a copiotroph system, and as we can see, the actinos would be considered more an oligotroph. If we look at the, I forgot to say on the last, these are high throughput sequencing data on MySeq, the bacteria are 16S, and the fungi are using ITS probes. So as we look at the fungi, same kind of map, there's a lot of information on this slide. I did kind of use shortcuts on the taxonomic titles here by abbreviating. I have the classes. I cut off the portion of the class name, the order name that are similar, just so that I could fit more information on the slide. These are all members of the Ascomycota. They were by far the most dominant this phase. Later in curing, you do see some Basidiomycota, and they do become more prevalent, but Ascomycota are really the dominant players. At the end of the thermophilic stage, we see that, yes, indeed, there are thermophilic fungi present. When you see the name like Thermomyces, that's a thermophile for sure. And it's also interesting to note that the abundance was much higher in these thermophiles in the aerated static pile, which stays a lot hotter for longer than the other methods. Later in the succession, then the community becomes dominated more by fungi that are more involved in decomposing wood, which is not surprising. So let's move on to nematodes. This is an interest that both Hannah and I share immensely. Nematodes, I'll just say we've both been working on using nematodes as biological indicators in other systems, but we wanted to see if they really worked and test them out for compost. So this is work that Hannah was involved with, and when she looked at compost, basically what you see are three kind of functional or feeding groups, and if you want to think about what they eat is one way to classify them. On the left-hand side are members of the bacterial feeders or bacteriivore groups you saw. They were relatively abundant, as well as fungivores, and then lesser abundant were these bacterial feeding predators. These were the only three trophic groups found there. There were no plant parasitic nematodes in there, but with these functional groups, we're able to calculate a fungivore to bacteriivore ratio. And she evaluated the community composition and looked at successional patterns across three different types of composting systems to see if she could detect any general patterns. The three systems are basically on the right is a barrel system, which would be more a small scale like you might do in your own backyard. It might vary from your own backyard in terms of they put all the materials in to the right C/N ratio and then closed it up and let it continue to compost. Where at home you might be adding material on a daily basis. In this case, they did not do that. So it would be a more controlled system. The two on the left are industrial scale. On the far left is one that is very, they're designed recipes that the Belgian government was evaluating and testing with farmers as kind of tools to promote organic waste management, but they're very precise recipes. And the one in the middle is what we're calling kind of the industrial green waste, and this would include all the landscape waste from household owners. So it's very heterogeneous, and you don't have as much control. There was some attempt to make adjustments, but it might not have ideally, met the exact carbon to nitrogen ratios at all times of the year. So, cool results. Nematodes really showed nicer, clear patterns more than even the microbial and the fungal communities. And I attribute that to they're a little higher on the food chain, so they're regulating more processes. They're feeding on those microbes, but they're doing much more. So on the left-hand scale, we have temperature, and across the bottom, you can think of that as time. basically, as you go through the process. So because the temperature this is starting with the thermophilic at the highest temperature, and as it's cooling down through the curing process and the maturation process. And we clearly see ecological succession. As you see in the high temperatures, it's basically dominated by the opportunists, kind of the R strategists, the bacteriivores. And then as the temperatures begin to cool, the food web becomes more complex. So secondly, we start to see the predators of those bacteriivores. So you see that combination. And then you start to get your fungal and bacteria feeding, and eventually the predators. But the really cool thing about that is that was a universal pattern observed in all of those cases where the compost was made properly into the recipe. There was a little bit more inconsistency when we looked, looking at that green waste where there was a lot more heterogeneity in terms of the materials going into the compost. So that pattern held for 5 farm compost processes, as well as the barrel process, the smaller scale. So this was very encouraging. And then then if we wanted to plot this and look directly at how the community index, and one of those is the ratio of the fungal to bacterial feeding nematodes, and look at how that ratio changes with temperature through time, you can see this negative relationship where that fungal to bacterial ratio is increasing as the temperatures are cooling. So that shows that can track and tell us something about the stage of maturity. The next phase was to try to take all this information from all of those processes and develop an index that could be standardized and used across different regions and different systems. So what Hannah did is developed this, trying to get some she set some criteria. One is to have repeatable and predictable patterns of succession, which I just showed you, to quantify the relationships between the nematodes and the abiotic parameters, and thirdly, to collate all the data from well-made, mature compost to that isn't, as I'd call, poor compost or immature compost. The criteria then were that if you wanted to define what a mature, basically then she could look at the entire continuum and then look at the top quartile, top 25%, and call that good compost or the mature compost. Okay, and when we look at those values, basically that translates into a nematode abundance of more than 500 individuals per 100 grams of soil, a fungivore to bacteriivore ratio exceeding 0.2, that's the lower threshold. They can go up to 1. The number of you had to have at least 1 fungivore, otherwise you wouldn't even have a ratio that would be higher. And also the presence of the diplogastirids. So the diplogastirids are really kind of a key. If you needed a sentinel species or taxa, that would be it. And then based on that, this is the scope of that project where she computed these indices, and she standardized the scales so that you could go across these and get them on traditional scales of like a continuous scale, because each of these kind of metrics has its own scale, and she wanted to be able to put them together and sum them up. 3.7 is the maximum value. That's the top line there. That's of the index. It goes from 0 to 3.7. And basically applying the same top quartile rule, values of 3.0 above are considered to be mature or finished compost that has the beneficial functions. So you can kind of see the group there at the top. Those represent the examples from the farming system, the case where they have very designed experiments following the seed-in ratios. They all reach maturity. And then on the lower part with the dashed lines are some that there's a little bit more variation. You can see the index values got as low as 0.4 on the index there, and that's translating to the green waste, which tells us it does matter how we make up that recipe. And that was really an immature and didn't have a complete soil community. Now I want to, okay, and if you're interested in calculating this, there'll be an online calculator available. It's being put online through University of Ghent. I think it's not up yet, but it will be shortly. Okay, so I've covered, we have a nice successional status, or successional progression of communities, composition change through the phases of compost. Now I want to take us out to the field and look at an application or use of compost to provide an ecological service. And in this case, I'm interested in looking at potential use for disease suppression. This work was based or built on really pivotal work that Harry Hoyntink performed in the 1980s at Ohio State University, where they were looking at nursery plants in pots and various compost maturity. And based on their work, basically they said, confirmed that they were getting better disease suppression with compost that contained hardwood bark and those that had maturity, more so than others, and that there were some composts that would be conducive. So we wanted to kind of test this idea in Vermont on a very prevalent disease on brassica crops, looking at early blight. And so we compared the treatments that I had the recipe for earlier in the presentation, compared 3 compost treatments with a Bayer. We had the control, which was a manure silage, and then I'll just present hay and hardwood as the 2 sources of carbon, and those were, designed to follow the carbon to nitrogen ratio and monitored. We conducted the study at 2 different locations for 2 full field seasons. And these are the data. These are disease severity data. You can see symptoms of the early blight down in the right-hand corner. We basically enumerated all the lesions on every leaf, and these were summed and averaged as numbers per plant, and we did that each time. And that's what disease severity represents. Each of the colors represents a different compost recipe. And on the left-hand side is site number 1, and the right-hand side is the second farm, and then the 2 rows represent the 2 years. You can see that one is truncated in the middle of a season there on the top row. Well, we had a tropical storm come through that day, and that entire field was under a foot of water after that storm. So that's why that experiment terminated. It's always the fun of field experiments. One of the things that I find so on the left-hand side with Riverside, if you see the black one is the bare soil, so that's no compost. And our control is basically the blue. You see a lot of noise, it's variable. This is the real world. But there is a trend for the hardwood compost to actually have less disease, suppress the disease more than the other compost types or compared to bare. In the second site, we were only able to differentiate with or without compost, statistically, but less disease with the use of that compost than without. So based on some other manipulations we did, we basically, we're not adding any biocontrol. The concept here is the compost is providing the reservoir or substrate for natural biocontrol to come in. So I want to just, to conclude, indicate that basically we feel like the curing is critical if we're going to really get the full benefit of compost, the biological benefits. And we're, on the disease study, that was with a fungal disease. We're expecting that there's no one compost that fits all, that we're going to have some designer compost, and there'll be some specific recipes and processes that match different diseases. So, for example, we're looking at how well it performs on a bacterial disease, Xanthomonas, at this moment. And from the opposite approach, Hannah's testing applications or actually inserting or adding biocontrol agents like BT or entomopathogenic nematodes into compost to see how well they survive. So we're kind of approaching it from 2 different angles. And with that, I'll entertain some questions.
question there, up there, up.
Hello, I'm here, the last line.
I can't see.
Yeah.
Oh, there you are. Okay.
I noticed that the suppressive effect of the compost you apply to the soil is not so good. So my suggestion is that if you can inoculate the compost with the different functional microbes, and then you will find the much better results to the suppressive the soil borne disease.
Well, that's one approach. I think there's two approaches. You can either, try to engineer or add the organism, or you can try to modify the environment so that they will come. And so this approach was the latter, in terms of trying to modify the environment so they will come.
Okay.
And yeah, I agree, it's variable, but I worked with exactly what the farmer did. I didn't manipulate any of their things other than, they could cultivate when they wanted to, and they planted different cultivars in different blocks. They're planting every 2 weeks, et cetera. And I guess I agree it's noisy, but I'm impressed that we're able to get that trend.
Yeah.
I mean, this is 4 replications, 2 years. I mean, that's not very many replications. So, you have the statistical power issue. So you might say it's messy, but I think it's the real world, and it's very economical.
But we found that if we just apply the compost to the soil, we cannot manipulate the microbiome of the soil. It's very difficult. So my suggestion is to apply the bioorganic fertilizer, not the compost, to the soil if we want to manipulate the soil microbiome.
It can be debatable. I think that's one philosophy.
Okay. And second question.
You have to also realize that you're going into a natural system, and if there hasn't been a lot of success in the literature on just introducing biological control species, because it might be entering, the niche may already be occupied, or it's added in a way that it has very low efficacy or survival. So, it's just different approaches.